Welcome back to The Explainer. Today we're diving into a genuinely fascinating dual breakthrough in the world of sensory neurotrauma. Specifically, we're tackling a condition that is just notoriously difficult to treat: blast-induced hearing loss. Now, if you follow neurological or audiological research, you already know that repairing the delicate microscopic architecture of the inner ear after an explosive shockwave is literally one of modern medicine's greatest challenges. It's like trying to rebuild a ship in a bottle after shaking it. But a recent study has unveiled a highly innovative, two-pronged approach to finally solving this. We're going to explore how researchers combined a surprisingly accessible blast model with a microscopic delivery system to not just treat but actually heal the echo of an explosion.
To really grasp why this research is an absolute game changer, we have to start with the human stakes. Consider this number for a second: 4.7 million. That is the number of U.S. veterans affected by service-connected hearing disabilities as of 2024. It is actually one of the most prevalent service-connected disabilities in existence, and much of this staggering burden comes from repeated exposure to high-intensity impulse noise, things like weapons fire, artillery, and explosive blasts. We aren't just talking about a mild, temporary ringing in the ears here. We're talking about permanent, life-altering sensory loss that currently has incredibly limited treatment options once the injury happens. So that brings us to the core question of today's explainer. Why is an explosion's echo so incredibly hard to cure? Well, researchers are basically hitting two massive, frustrating roadblocks. The first one is on the engineering side. Explosions are incredibly difficult to replicate affordably in a lab. Traditional methods require million-dollar shock tubes or massive, heavily armored blast chambers, which severely limits how many scientists can actually get in there and study the problem. The second roadblock, it's biological. The inner ear is essentially a biological fortress. Even if we had a miracle drug to cure blast trauma right now, the inner ear is physically locked away from treatments by tight cellular barriers and the eardrum itself. You can't just swallow a pill and expect it to magically reach the precise microscopic structures of the cochlea in high enough concentrations to heal the trauma. Okay, so how do they get past that first roadblock? It's actually kind of genius, what I'd call a real meta breakthrough. Instead of relying on the old way, those prohibitively expensive shock tubes that almost nobody has access to, they developed a totally new way using something completely unexpected. They cleverly modified a commercial paintball gun device connected to a compressed air tank. Seriously, a paintball gun. By repurposing this highly accessible, off-the-shelf recreational item, they created a highly reproducible, cost-efficient model to study blast trauma. This completely democratizes the research, allowing labs pretty much anywhere to study blast injuries without needing specialized, military-grade infrastructure. And this right here brilliantly illustrates exactly how they optimized that paintball device for auditory research. If you look at the pressure graph on the right, you'll see the blue line for the modified model is much lower. By physically widening the device's outlet window from 6 millimeters to 25 millimeters, they significantly lowered the peak output pressure. But check out the graph on the left. The sound intensity drastically increased. So, by just tweaking that outlet size, they perfectly mimicked the key characteristics of a real-world explosive blast, delivering that massive, damaging wall of sound, but without an overwhelmingly lethal pressure wave. It gives them a totally controllable, reproducible injury model to work with. All right, so the engineering problem? Solved. But we still have to breach the biological fortress. Like I mentioned earlier, actually delivering therapeutics to the injured inner ear is just a monumental hurdle. Under normal circumstances, the tympanic membrane, what we all just call the eardrum, stands as a physical barricade, completely blocking access from the outer ear canal into the delicate middle and inner ear structures. Systemic treatments, like an IV or a pill, well, they just get filtered out by the body before they can reach the ear in meaningful doses. So the question is, how do you get the medicine exactly where it needs to be, precisely when it needs to be there? Here's where it gets really wild. The researchers found a brilliant, almost counterintuitive solution. They used a silver lining approach. Step 1, the intense blast wave itself actually ruptures the eardrum. Normally, that's just considered collateral damage, but here, they used it as an open door. Step 2, immediately following the injury, while that hole is still literally wide open, researchers inject the therapeutic nanoparticles directly through the ruptured eardrum, bypassing that barrier entirely. Step 3, nature takes its course. The eardrum naturally heals and closes up on its own within about a week or so, effectively trapping the medicine inside the inner ear where it can do its work over a prolonged period. It is an incredibly elegant way to turn a traumatic injury into an immediate delivery vector.
Let's look at how this builds into the actual treatment. What exactly is going into that temporary hole in the eardrum? Well, the researchers didn't just squirt raw liquid drugs into the ear. They used PRGP nanoparticles loaded with a molecule called 7,8 DHF. Let's distill that jargon for a second. Think of the PRGP nanoparticles as microscopic, biodegradable delivery vehicles. They're specifically designed to bypass cellular defenses, slip right into the cells, and then slowly degrade over time. Inside these little vehicles is the payload, 7,8 DHF. This is a specific molecule that acts as a TrkB agonist. In plain English, it artificially triggers a naturally occurring neuroprotective signaling pathway in the brain and ear. Instead of just masking symptoms, the slow-release payload literally signals the damaged cells to survive, maintain their connections, and heal. But the big question, does it actually work? Let's follow the data narrative. These squiggly lines you're seeing are ABR waveforms, essentially a way to measure the brain's electrical activity to see if the ear is actually hearing anything. Immediately post-blast, if you look in the middle column, both the treated and untreated groups just flatlined, no hearing. But look at the results 28 days out on the right. While the untreated green lines remain totally flat, the purple lines, these are the mice that received the nanoparticle treatment, show distinct waveforms returning. Their functional hearing was restored remarkably close to their original baseline. The treatment functionally rescued their hearing. It's amazing. Now, functional recovery is fantastic, for sure. But in science, we need to look under the hood. We have to shift from functional recovery to structural recovery. If the animal can hear again, we need to look inside the microscopic architecture of the cochlea to ensure we aren't just treating some superficial symptom, but actually preventing the physical death of the ear's critical sensory components. We need to see if we're rescuing the ear's built-in amplifiers. And this is where the histology visuals become absolutely stunning. What you're looking at in neon green are the sensory hair cells of the cochlea. We are specifically interested in the outer hair cells, which act as the physical mechanical amplifiers for the ear. When an explosion happens, the shockwave violently destroys these cells, and once they die, they do not grow back, ever. In the middle row, the blast group with no treatment, you can clearly see massive gaps and missing clusters of these neon green outer hair cells, especially in the basal region of the ear. But look down at the bottom row. This is the blast group treated with the 7.8 DHF nanoparticles. The rows of outer hair cells are beautifully intact. The targeted, slow-release treatment physically stopped these amplifying cells from dying off after the blast. Okay, so preserving the amplifiers is vital, but there's one final, absolutely crucial piece of the puzzle. Think about it. What good is a high-end amplifier if the cables connecting it to the speaker are totally severed? In the inner ear, these cables are the inner hair cell ribbon synapses. These microscopic synaptic connections are the crucial wiring that actually takes the sound information and transmits it to the auditory nerve and straight up to the brain. The impact on this wiring is crystal clear in the data here. Blast waves are notorious for causing cochlear synaptopathy, meaning they literally shred these delicate synaptic connections, even if the hair cells themselves somehow survive. The green violin plots show the untreated blast group experiencing a massive, devastating drop in the number of these ribbon synapses, especially in the highly vulnerable basal region of the ear. But look right next to it at the purple data. The group treated with the localized nanoparticle delivery successfully maintained their synaptic wiring. The number of connections remained statistically comparable to ears that had never even been exposed to a blast at all. The treatment preserved the whole communication pipeline to the brain. So let's pull back and look at the big picture. We've seen how a repurposed, off-the-shelf paintball gun allowed researchers to unlock the mechanics of blast trauma, and how using the injury itself as a doorway allowed biodegradable nanoparticles to physically save the inner ear from permanent deafness. It's just an incredible triumph of out-of-the-box thinking. But it leaves us with a profound, much wider question to ponder. If we can design microscopic, slow-release delivery vehicles to successfully reverse severe blast trauma in an environment as complex and isolated as the inner ear, what other neurotraumas could we treat next? Could this exact same localized delivery framework be adopted for traumatic brain injuries, optic nerve damage, or other locked-away regions of our nervous system? The echo of the explosion might finally be fading, but the impact of this delivery system, it's just beginning to resonate. Thank you so much for joining me for this explainer, and as always, keep learning.